So welcome to the RISE ECE Distinguished Summer Seminar Series. And this talk is also co-hosted by the IEEE SSCS Distinguished Lecture. So today, it's a great honor for us to welcome Professor Dejan Marković from UCLA ECE Department. So Professor Marković is currently a professor of ECE at UCLA, and he is also affiliated with UCLA Bioengineering Department. He received his PhD in 2006 from University of California, Berkeley, where he received the David Sacrison Memorial Prize. His research is at the intersection of computer engineering and bioengineering, and currently his work is mostly focused on implantable neuromodulation systems and domain-specific computing architectures and design methodologies. Professor Marković has received a number of prestigious awards, including the NSF Career Award, and he also received the ISSCC, which is the top conference in the circuit design field, the Jack Draper Award for Outstanding Technology Directions, and he also received the 2014 ISSCC Louis Winner Award for Outstanding Paper. So during the talk, please use the Q&A function at the bottom of your Zoom window to ask questions. And I will allow you to talk, or I will read your questions by the end of the talk. So if there's nothing else, let's welcome Dejan to give the talk.
Okay, well, great. Thank you, Karyan, for your kind invitation and introduction. It's a great pleasure to be with you. I just wanted to say hi. This is a virtual hello from a real person, and I'm going to mute my video for bandwidth reasons so that we can focus on presentation. Today, I'm going to talk about recent work and overall perspective on closed-loop neuromodulation. There is accumulating clinical evidence, and everything starts and ends in clinic when it comes to the need for closed-loop neuromodulation. There was a study in a 4-patient Parkinson's study that shows that equivalent outcomes can be achieved when stimulation is only applied 45% of the time. There was another study from UCSF, from Dr. Starr's group, showing again 2-patient Parkinson's disease study that shows that with closed-loop neuromodulation, you can have the same outcome with 40% less energy if you judiciously apply stimulation. And obviously, these results state that you can essentially be more efficacious in the way that you deliver therapy by introducing, recording, and closing the loop. Less energy that is introduced into the system could potentially lead to less side effects and more efficacious monitoring of the condition, as well as better therapy. Dr. Lozano is also prominent in this area, kind of outlining the need for closed-loop DBS in Alzheimer's patients, and particularly when it comes to cognition and memory issues, there is even more specific need to do stimulation at a certain phase of the waveform that is correlated with encoding or retrieval of memories. There was a large recent study with University of Pennsylvania under the DARPA RAM project that has shown interesting and promising results on closed-loop DBS for memory and also TBI. So when you put all of this together, you pretty clearly understand that existing therapeutic technology has significant limitations that prevent clinicians from exercising more degrees of freedom and getting more advanced closed loop features. So from these clinical needs, then we can motivate and understand the following technology needs. First, we need to do sensing from a high number of channels, desirably, for indications that are governed by neural networks, not just single focal points with a small number of channels. Secondly, when you speak with about closed loop, then you would want to have full duplex sensing and stimulation to go away from walkie-talkie technologies and get to really full duplex technology without blanking stimulation, recording during stimulation. And that's particularly true if you want to do this kind of phased locked stimulation methodologies. Then real-time wireless data is really almost essential when it comes to recording and having a full access as needed to monitor brain activity for diagnostic purposes and further to inform closed loop. And it turns out that this would be megabits per second of data if you have a large number of channels. And then, of course, these increasing technology requirements wouldn't be challenging as much if you were not to add miniaturization and higher channel count that comes kind of conflicting that requirement, because less is more when it comes to implantable devices. Less invasive is better, so you need to take that into account. I like to also point out that to give honor the legacy of UCLA as the founding place for brain-computer interfaces, and this is also instructive to consider when it comes to the development of new technologies. In the 1970s, in 1973, in the computer science department, Professor Jacques Vidal introduced the idea of brain-computer interfaces and coined the term BCI, and laid out a foundation for two-dimensional navigation task, which is the bread and butter of BCI and neuroscience, both basic and translational science. So you can see here roughly from his paper in the 1973 was the foundation of BCI lab, which had the experiment room with the subject manipulating a joystick and observing results on the screen. Then we had a control area where you have a signal recording, and then further computer area for processing, and then closing the loop with the display. So I mean, I like to use this because the technology that was available at the time was very distributed and fairly bulky to implement as compared to what we have today for this kind of relatively slow kilohertz rate application. It's also interesting to see here that the overall computer that was available for the whole campus was 4 megabytes of memory. And it was networked in this ARPANET, which was the first node of the internet that came in the 1969. So pretty major discoveries around that time at UCLA with the internet and the BCI. And in 1976, there was the first demonstration of volitional control of a two-dimensional maze, a navigation task through a cursor, and then BCI is about decoding algorithms and really interpreting that in a kind of the two-dimensional screen. So there was, just to understand the overall trajectory of this and the implications, in 2000, there was an experiment at Duke University by Professor Miguel Nicolelis that showed how you can, apply this robotic arm control in an animal and then transfer to another remote place at MIT Robotic Lab to control a prosthetic. And I like to say this is the application of both in UCLA discoveries, the internet and the BCI, to perform this kind of experiment. Then shortly thereafter, there was another experiment with the two monkeys, Ivy and Aurora, playing video game and at some point realizing that you can pretty much control the outcome with thoughts rather than the joystick, and they just gave up on the joystick and were able to continue with volitional control. And then translation to humans is something that everybody obviously is interested in. This was the recent example from Stanford University from Dr. Jamie Henderson and Professor Krishna Shenoy, where they've shown how you can type through this kind of visual interface and feedback for paralyzed subjects. This is the BrainGate technology that shows the BCI interface with these electrode arrays. I'm sure everyone's familiar with the Utah electrode array that you then connect to the BrainGate device. This was the original neural interface. This is a lot more miniaturized now, but nevertheless, you have a percutaneous technology that then brings the signals out to be digitized and decoded in a computer to assist in various kinds of 2D space experiments. I think there is a question here. Someone, here's a question. At least someone raised a hand, so let me. Okay. Maybe we will save that for later. And this just shows the technology emphasis on this and the time scales from 1973 concept to first in human application 2002. So that's about 3 decades. This is fast forwarding to just last year around this time with Neuralink have disclosed in their white paper the brain-machine interface technology that has a very impressive technology for robotic injection of high-density microelectrode arrays that are all placed on a flexible material, and then this machine then lifts them up one by one, puts it close to the needle that just injects them in there. So it's kind of really sewing machine that allows you to put hundreds of these electrodes in a very tight space of a few millimeters, which is a great tool for BCI. And of course, a lot of questions remain in regards to clinical translation and therapeutic use of this kind of technology. Now, when it comes to clinical translation, there was kind of a little bit of a detour to the BCI, but what I would like to get into is really connecting the clinical, scientific, and technology aspects together. And access to deep brain areas where you have the ability to record a large number of electrodes for tracking of the epileptic seizures is really uniquely affordable in epilepsy monitoring. These are patients who are refractory to medications, and they're implanted with these, in this case, I think it's 8 electrode arrays that have about close to 100 contact positions all over the brain. And they are recorded for about 2 to 3 weeks to track seizures. And the end of it, they are then, if successfully localized, subjected to resective surgery. If not, all of this is explanted, and then another device is implanted to try to do a treatment of seizures. People also use this environment to be able to record even single unit activity in a human brain. And shown here is the electrode that is built at UCLA by Dr. Fried and Eric Benke, that is called Benke-Fried probe. They take essentially LFP electrodes and they insert a number of bundle of microwires that allows you to observe activity of single units. And when you essentially look into these anatomical images, then you see that some of these areas in the medial temporal lobe in the epilepsy patients coincide with areas that are interesting for memory research. And in the periods where patients are not seizing, which is what clinicians would like to do, then scientists come and do various experiments to study human memory from these areas and be able to develop translational theories and kind of put this into therapeutic translation pathway. This is still, open loop, largely with very limited closed loop, and the technology that are very bulky bundles of wires that are connected to these instruments for monitoring of the intracranial waveforms, as well as the patient behavior as they're subjected to these experiments. What's really interesting about human memory is that, unlike motor cortex, which is the kind of the driving application for BCI, human memory is non-topographic, meaning that cells that are right next to each other could encode entirely different concepts. So you can see here one cell, just for illustration, encodes a movie character, another cell records something entirely different. And people have done a lot of experiments to find out that there is also associations, like this cell here responds to associations of the elements that could broadly be classified as, sea world, like a boat and a fish, for example. And this came out of a lot of experiments really trying to access these fine features of the single unit activity that Professor Fried and his group have conducted over the years. And so this is one example here to show single unit responses which fall in the audible range. So if you look into the spikes, they are somewhere in the audible range between 300 hertz and 5 kilohertz, so you can even hear them. And so I'm going to see if this is going to play here. I just need to get to my exit the laser pointer. How do I do that? Over here, arrow. Okay, I think. Okay, there we go. I think I got my clicker over here. So you may or may not have audio that comes with it, because I don't think I've been able to share computer audio, but you can see the response of one single cell, that is the amygdala, and you present a variety of human interactions in various settings. This is meant to show that this neuron only responds to very time couple and nothing else. And then over time, people have determined that it also responds to characters from the TV show, which gave rise to theories associated with and association properties as well. And then again, you go into various other kinds that neuron absolutely does not respond. So it's a highly selective neuron. So there is, interesting translation opportunity for human memory, but there is a lot of technical challenges that are left to be resolved with respect to the longevity of single units, the specificity to which you can access memories, and many others. So just looking then into what is realistic today and the technology that's available for therapy, this is the device that is used in the Parkinson's disease and movement disorder indications. The overall idea originated in the 50s, and FDA approval came in 2000s. So just kind of setting realistic expectations for new technology, which typically takes a long time for full market use. So this is a well-established surgery that is nevertheless a long procedure. You can see a device that is, believe it or not, adapted from cardiac pacemaker that then runs the wire through the neck and drops down the lead for open loop stimulation. This is not closed loop. So this is open loop stimulation 24/7. And it is common surgery. Like I said, it's been routinely performed all over the place. These days, about 10,000 patients are implanted every year. And it happened to be, In a few years ago, we had 500 patients at UCLA. It was a milestone surgery that was then broadcasted through the popular online media to kind of increase the public awareness of the surgery. And I'll just show you a piece of it that happened to be a famous musician who was patient number 500 over here, that just shows how this surgery is done, that you have these stereotactic frames for precise positioning of the electrodes, and it's a wake surgery where patient needs to kind of help doctors find a sweet spot for stimulation. And once you lock the position of the lead, then you go for a second stage surgery to put implantable pulse generator in the test and hook up the lead to the IPG. Now, if you look into across the board, you see that there is an increasing burden of neurological disease. If you just see, for example, the cost of managing over here, let's see what happened. The cost of managing Alzheimer's, chronic pain, depression, mental health in general exceeds $1 trillion per year in the US alone. And while deep brain stimulation has been efficacious for Parkinson's disease and very limited efficacy for epilepsy, where you can see that despite these numbers, you only have about 300 implants per year, there is virtually nothing that's available for these technologies, indications which are essentially at a scale that is beyond what these tools are able to support. So these indications are involving brain networks where you have many recording sites and many stimulation sites in order to address this indication at the right scale. So simply this old technology from cardiac pacemakers is not going to be successful in treating these indications. And people have actually tried. There were failures or limited success, I would say, in treatment of depression. Alzheimer's trial is currently going on in phase 3 that is not showing promising results. And even the technologies like this, which is technology-driven work from the neurospace, fundamentally is still a four-contact interface with the brain. From the clinical opportunity, it provides very limited benefit. And so this goes back to my earlier comment that access to these networks is really affordable in epilepsy monitoring, where we can access many electrodes and be able to create translational science for these network scale indications. There is a major lack of technology when it comes to treatment of these network scale indications. Deep brain stimulation is efficacious in Parkinson's, and neuroprosthesis is also efficacious in the treatment of deafness with the Cochlear implant, because the tools here are the right scale. You have a focal stimulation of subthalamic nucleus or stimulation of the cochlea, but all of these other indications, which are much larger in the number of people. The numbers here show the number of people worldwide with these indications. So if you look into, for example, depression and Alzheimer's, that's 50 times larger number of people than Parkinson's disease, which is the bread and butter and the driver for the DBS industry today. So without proper tools to diagnose indications and without proper technology to close the loop and do therapy, there isn't much progress made. And you also need to account social issues and many layers of how this technology comes into the society and what are the obstacles. Like in the cochlear implant, you may be deceived by relative success, but it takes about 10 years for people from the time they're told to have a cochlear implant to actually adopting it, because of the obvious intrusive nature of this and the big social mark that everybody can see for these patients. And if you're trying to translate something like this to mental health, it will never go much further. So anyway, there is a lot of issues here, and technology really needs to come to the right scale to treat these next generation problems. So imagine that you can then record from multiple locations of the brain and stimulate to self-adjust and optimize therapy that is individual to every patient, that takes into account anatomical differences as well as the brain behavior that can be adapted over time. So in order to build a prosthetic, it's instructive to look into these previous technologies such as cochlear implant, very successful. It has a microphone and the processor in the external piece that then sends the stimulation pulses through the inductively coupled power and data link on the implant that then delivers the stimulation in the cochlear, which then transfers the stimulus to the auditory nerve and reaches the brain. So this requires also alignment magnet over here, which is similar to the technology you have also in your electronic toothbrush to handle the sensitivity to the displacement of coils on the primary and secondary side. Retinal implant is perhaps the most challenging technology, and it still has a very low traction due to various limitations of the benefit to the patient. But if you look into just miniaturizing the implant to be able to be attached to the retina, solving the issues of the detachment of these flat arrays to the retina, getting the RF coils for the wireless power transfer when you have to deal with a constant displacement of the coil with respect to the primary coil over here, and dealing with the miniaturization of this in the packaging to deliver the stimulation is just the incredible level of technology difficulty that people have to go through. And this example shows technology from Second Sight that brought this technology forward. Now, this is where government also comes in to try to accelerate development of technologies that would try to really become more clinically efficient and sustainable. If you look into historically what Department of Defense, DARPA particularly, was interested in these kind of EEG caps for a variety of applications, of course, that are multi-purpose for military defense and generally for consumer applications, the DECA arm, the brain-computer interface, which was an NSF project first and then the DARPA project. When you see across the board here, these technologies are not really nicely embedded in the lives of people. So technology has got to be minimally invasive and provide benefit to the patient. And that's where President's Brain Initiative came to develop complex understanding of the complex brain disorders. There was a program for restoration of active memory and also a program for mental health issues in military, where government recruited large teams of neurosurgeons, clinical scientists, basic scientists, and technologists to come up with the next generation R&D prototype that is capable of providing up to 200 or more possible interaction points with the brain to be able to interrogate brain networks as compared to 4 to 8 contacts, closed loop, wireless technologies, and so on. So I'll share some results with you from the development that my team was involved with together with UC San Francisco, Dr. Eddie Cheng, who was the PI on this project, and we had Lawrence Livermore for packaging, Boston Scientific, and a large number of collaborators to make this possible. So we envisioned a system in which we have a modular system, which is for R&D purposes only, for recording and stimulating the brain, then aggregating the signal, and then the control module for closed loop online control with wireless technologies to program the device, and also wireless data telemetry, as well as the overall user interface for convenient selection of the electrode channels that clinicians can program the device to access, and wireless power and data. So the end-to-end end goal for this was to develop a really a system that can then build on top of these devices closed loop algorithms that you can imagine, algorithms in the cloud that can be used to be tuned to individual patient, and then you have a patient in the loop with the kind of application that gives the subjective access and scoring of the mood to synchronize with the device and be able to do refinements as necessary. From the technology perspective, you go into the hardware lab and you quickly find that everything is really clinically driven and everything is system driven. So any idea of chip design that you want to build and publish a paper on just falls off the cliff. So here we put the infrastructure to develop all of these elements for example, stimulation and power management, sensing. We got to put a control and have the ability to control it from the computer, to write a lot of embedded code for it. Then we had a wireless power delivery here over these pair of coupled coils that then powers this setup, so that you can demonstrate any component in the context of the complete system. So once you pass that stage, you go to the next level of miniaturization, which is convenient for taking this device as a trial device. You can take this to the bedside and do clinical experiments for things that you cannot do in vitro with PBS, but really you need a human brain in the loop to be able to do this. And once you graduate from this level of complexity, then simulation and sensing, you'd go finally to the implant scale, where the challenges for assembly, system integration, and testing are much harder considering the limited access to these devices. And so we built a system that can simultaneously sense up to 256 points, aggregate data, and go down to the control module, and the controller then in response sends power and therapeutic stimulation that is individual to each patient. This technology features the world's smallest human-grade requirements neuromodulator that can provide full duplex 64 contact sensing and stimulation. You can see it fits roughly in the size of a vitamin pill. This is a 12-layer board which has 50 micron traces and spaces, lots of blind vias, and from the assembly standpoint, it presents really, really incredible experience building custom pogo pin sockets to debug this and dealing with the mechanics of this. Then layering up chips. Here we have a power and stimulation engine. We have a sensing chip. We have crystal oscillator, and the rest is medical grade capacitors for power delivery. And then you have to put this on a ceramic substrate and then do the hermetic laser welding for the package, and then need to communicate with this whole device with 6 pins only: differential power, ground, clock, serial in, serial out, and validate a large number of components here. Then we have the controller module, which is leveraging the packaging technology from Boston Scientific that we built, then the printed circuit board and the controller and the wireless technology that was custom developed that fits in this enclosure and passes multi-point inspection test for industry grade neuromodulation system. So this was taking a number of revisions to make sure that full compliance with respect to mechanical and electrical subsystems, as well as these connectors, are ensured at the manufacturing facility. These are the electrode arrays that are built at Lawrence Livermore National Labs, and it's equivalent of throwing away paper map and using Google Maps for navigation when it comes to higher density access to the brain, and going away from 4 contacts, which is kind of continent level access, to 64 contacts per lead. So we have both cortical leads as well as depth leads. So these are 4-way split contacts that allow us to do current steering and very complex stimulation patterns. This system was meant to be modular, where we can put any number of cortical or subcortical leads for R&D purposes. But of course, for real therapeutic translation, there is a lot of elements that you don't want to put into such a system. And this is just early demonstration of the capabilities. I will show you here quick video of the electronics subsystem that is the first generation neuromodulator that you will see the overall infrastructure taken for measurements. So you have to have signals that you inject in the beaker. Then you have the developer version control software that allows you to observe waveforms from this custom electronics on the computer, as well as to configure the stimulator that you can see here, the output of the stimulator. And then you have the tiny module that is in the pogo pin socket over here for testing. And this is FPGA and microcontrollers that have embedded code that burn the configurations on the neuromodulation unit and interfaces with a computer to display the results. So here we first inject a signal into this large reference electrode. Then we perform stimulation, which is about 10 volts pulses, plus minus 5 volts. And then we are doing recording from the contacts of these electrodes, which is shown here flattened out because it's a MEMS fabricated electrode. Then you were supposed to catch that recorded signal, of a reference signal, in presence of this large stimulation artifact to measure what is the dynamic range necessary for simultaneous sensing and stimulation, and also be able to demonstrate the operation of this circuit. You see here the configuration interface for taking 4 stimulation engines and mapping them up to 32 contacts. So you can see here the amplitude programming, then you have the pulse programming, and then over here we can put any arbitrary waveform. I wouldn't say arbitrary, but I would say custom. You can customize waveform to put any kind of discrete approximation for various harmonics that you want to put in here, and it is all charge balanced stimulation. So you can see that the actual waveform shapes change on the oscilloscope in these discrete levels that approximate these desired waveforms. Then once you are able to get recording in presence of stimulation, we've built online algorithm for stimulation artifact rejection that first determines the signal statistics based on baseline activity absent of stimulation. And then it learns the signal propagation from the stimulation site to the neighboring recording sites and uses that mutual information to clean the artifacts. And you have to keep in mind that this is a time-varying nonlinear mapping. So it's important to understand how to develop filters that deal with these dynamics and nonlinearity. You can see here the plots of signals in time and frequency domain without artifact rejection on the left, time plots and frequency plots, and on the right, clean signal in both time and frequency. And of course, the difficulty here is unavailability of the ground truth, and how can you really reliably do this kind of artifact cleanup. In order for us to understand what's required for dynamic range, we take clinical experiments to inform technology development. So here you see one of these Benke-Fried probes from a human epilepsy patient, and you are looking into right entorhinal cortex region here that has the two LFP contacts. The most distal of contacts, you're doing bipolar stimulation, and through that whisk of microwires, you're doing micro recording over here. So this is 7.4 millimeters apart from the these contacts. And then what we do is we increase the amplitude of stimulation and see what is the required amplitude on the recording site so that we can have a full signal plus artifact. And you have to keep in mind that when you do this with existing clinical equipment, those are wall-plugged instruments which have no power limitations. And we're trying to develop a technology that can handle this with implantable microwatt level power. So here is what I described. You are increasing the amplitude of stimulation 0.2, 0.4, 0.6 milliamps and further. And you can see in blue is the signal plus artifact. Okay, so I think we have a question here for the denoising. There is no ground truth. Is signal not generated from the signal generator? Yeah, that's a good question. You can put synthetic signals, right, for the development, but and you can in fact generate synthetic signals from the brain and put them through the beaker, right? But that itself does not capture the real brain in the loop, where you have the subject-specific time dependencies and non-linearities. That is something that you just cannot do in the beaker. And so when you insert your probes in the brain, then brain does, signaling the way it does it. There is no ground truth there. There is, a whole bunch of noise and things that you see from signals that are further away from the vicinity of the tip of the electrode, and there is just no way of knowing. You can synthesize all you want with the models of noise and whatever you can bring in, but strictly speaking, the only way to really reliably measure the responses in presence of stimulation is when you do that in vivo. And once you're in that environment, there is no ground truth, basically. Okay, so this shows that in this particular case, you have a headroom of about 5 bits. So this is the extra dynamic range that you need to support in order to provide simultaneous sensing and stimulation. Let me see. Okay, yeah, great. Yeah, thanks. So yeah, we can continue to discuss this. And so you need to then look into what's required here. And so we took a whole number of approaches here. We've developed several chip prototypes that were first working through the LFPs only here, where we took essentially time to digital conversion, realizing that increasing the number of bits in the voltage or current domain is very difficult because there is a fundamental trade-off of power and noise, which is very different from wireless communications. When you get closer to the base station and your signal is stronger, you can drop the gain, your noise gets higher, but signal to noise ratio is maintained. Well, in the brain, nobody amplifies your neural signal, and you have that in-band interference that you just need to deal with. So that's why we kind of try to escape from that current to voltage to voltage or current to voltage conversion, and ended up with this time to digital conversion solution, which provided up to 100 millivolt linear input range. And then we came up with other techniques to do 200 millivolt and deal with spikes that is referenced in these publications. One very important thing that people overlook is the need for input impedance. Okay, so this paper, I don't know if it's shown here, but Medtronic, famous chopper paper that everybody refers to, is great solution for 4 contacts. And the real, I think, you need to be real careful when you talk about human grade technology that you need to avoid electrolysis. And so you can easily put DC blocking capacitor to do that in a 4-channel system. But when you're talking about hundreds of channels, you just cannot do that in a miniaturized implant. So you cannot compromise input impedance. You need to do something that's different on the chopper stabilized amplifier. For science applications, it's fine. You can do this lower impedance, but you need more than a giga ohm impedance, and that's something that many people overlooked, unfortunately. So this is the resulting technology elements that we came up with. It's a large number of people. It was probably a team of about 50 people all together with clinicians and technology. And you can just imagine what level of fun you can have with the purchasing and getting everybody to be on the same schedule with regards to components. And it shows this distributed system of neuromodulator, aggregation module, control module. This was from the DARPA D60, 60th anniversary of DARPA. We had the exhibit to show the difference between the closed loop system that you see on the left when you activate stimulation only as needed, and the open loop stimulation on the right. The verification of all the components was also a very important factor. So we've developed this motherboard that is really providing all sorts of connector interfaces that allows us to test components at various levels of maturity. So here you can see Goldfinger frame for the CM module that after successful testing, you can excise the electronics from the frame, do the packaging, and then you have another interface that you can do fully packaged part. We've also built emulators as well as real parts. So you had a lot of flexibility to test the full system at various levels of maturity. And so we've got all the system components with a full communication chain, control from the PC application to do that. So these are the images from the last 5th-year review from DARPA program last year, where you can see the full system here in its real form with the electronics, including the chest module, the data coil for communications that was custom developed, the aggregation module, and we've built dozens of these for qualification purposes. And even the things that we don't even publish about, when you say that you had like 37 out of 47 units that passed, think of like a 50-page test report, which has 22 test verification points where need to be all manually signed by an operator with specific numbers to test variety of things that go before insertion of the battery, before the package seal, and dealing with a whole bunch of things that needed to bring all the realities of what it takes to build qualification units for a system like this. And we've spent most of the time miniaturizing this electronics, and nevertheless developed all of the other modules, including custom packages, connectors, cables, and all of that. Now, if you look into this system, it is still a bulky system because of the R&D and modularity request, and of course we would like to evolve this to a system that is more clinically viable, which takes a lot more resources and time to do, towards something that is much smaller volume and much more integrated, taking all the lessons learned from the previous experience. So these are the references for that work, comparing here essentially technologies that are R&D technologies, commercial technologies for science use, basic science, and then medical grade technologies, which is really aim for this project was to pass the FDA and CE mark specifications. And then, like I said, we would like to take this to miniaturize and bring it as a unibody device with passive electrodes and be able to support things like temporary epilepsy monitoring and taking into account all the regulatory and the clinical business aspects and the pre-market approval. That's a whole another story, how this technology can be transitioned to something that's at the next level of maturity for the clinical world. There is a big neurotechnology landscape. When you start to do things like this, you better watch what's already out there, because people have spent a lot of time and effort previously, and you can classify these different technologies into neurosensing, neurostimulation, brain-computer interface, including the Neuralink and other technologies. And then you can see neurostimulation, Boston Scientific, Medtronic, and Nevro, all these other companies, and electrode manufacturers, and so on, to understand this whole kind of landscape of applications and think of it like a totem pole of technologies. Some technologies are diagnostic only, some technologies are just imaging diagnostics, but ultimately you want to provide therapy, so you have to have stimulation. And so then the question is, what else do you need to have to close the loop and inform this whole stimulation strategy? And then the space becomes kind of starts to declutter once you put those constraints and see where the opportunities are. So this is something that we didn't put in our DARPA statement to work, but it was the most useful thing that we built, which is this external device, which I call Cracker Stack, because it's literally the size of a cracker, and you can put this together like a neuro Lego approach that, even, preschool kids can put this together through snapping the connectors and telling them how to arrange the colors. So you have a 64 contact stimulation and sensing using the chip technologies, which we miniaturized to a lot greater extent. The signaling layer and power delivery, which comes through the micro USB, and the FPGA for embedding the code and running the closed loop. We've also built a graphical user interface, packaged this clear with a see-through that you can see here, but also packaged through the copper tape and shielding against 60 hertz to be able to take this to clinical experiments. We got the IRB approval for this to run on human subjects at UCLA. We've done recording. We are now doing stimulation to get the stimulation response maps and be able to then go back to the drawing board for refinements of the stimulation artifact rejection and other things that we want to support in the future. These are some of the examples showing recording of neon codon clinical instrument, and our device that you just saw on the previous slide that shows the time and frequency plots that practically do not reveal distinguishable differences between these two systems, which is meant to show concordance of our device with the clinical system. And we've done some stimulation that's been sort of now on hold a little bit because of the COVID situation, but we'll be going back to it and we'll have more clinical experiments. I've also ran some interesting work in the neurotechnology to kind of connect this with non-invasive methods, and because the screen sharing might not, the sound sharing here may not work. I don't know if I enabled it, but this is the concept of brain stethoscope, the device that we called brain stethoscope. At the time, I was working with colleagues from Stanford when I was on my sabbatical, Dr. Parvizi, who is a neurologist, and Chris Chafe, who is a computer musician. And I brought my neuroengineering expertise to come up with the electronics part of this to essentially take EEG waveforms and convert them into sound. And this is the speaker that converts the sound of the EEG into, the EEG waveform into the sound. And you can literally put a stethoscope for the heart on it. So we called it initially brain stethoscope. This became the founding technology for Cerebell, which is the rapid EEG response that is device looking very different from this one. It has a screen, it has the sound, it has 8 contact points, which is required by FDA for 510K device. You can look up on Cerebral website to kind of see these cool sounds and how easy it is to get from like no epilepsy, no seizure, background alpha activity, flat tone to more like earthquake type disruptive signal of patient with seizures. So this was a lot of fun. Kind of connects up to the epilepsy monitoring world. And if you points to take away here is that although it is very challenging and low volume kind of market for electronics development, there is a great opportunity and a great need in coming up with technologies that are capable of addressing mental health issues and other network scale indications, things like memory restoration and so on. And beyond medicine, it would be interesting with the data that you get from the deep brain structures to understand neural mechanisms of cognition and memory that could, in patients who already have primary indication for something else that justifies implantation, augment human learning and knowledge. I'm not talking about AI and stuff like that. I mean, that's to me a little bit premature to jump that far without really solving major medical issues to talk about, somehow controlling human brain with machines. That is just, I just can't see that. But getting like better memory and things like that and accelerating knowledge, I think that's realistic, and we should look into that. And then, people fantasize about also building artificial intelligence in machines, although I would, take that also with a caveat and realistic look that, society didn't bring airplanes by mimicking flapping of the wings of the winged animals. So we just built, jet engines and, solid body of the airplane, and it works. So we need to also kind of look into how to implement learning and AI that may not have just the same compute fabric as the brain, which is ultimately pattern processing machine. And so this is it. I mean, I wanted to share with you my experience in building technology, which meant to displace decades outdated technology from the cardiac pacemaker to something that's a lot more on the scale with capabilities of today's technology and the ecosystem that exists for data analysis. And I jokingly say, but half jokingly, we can no longer implant telephone poles in the era of smartphone technology. We really spend so much time innovating for consumer applications and virtually do nothing when it comes to health. And this is a strong message that I like to project to people who are in the policy making and investments and various kinds of communities that need to come together to enable better technology for medical applications and big unmet social needs. So I'm going to stop here and see if there are follow-up questions.
To the audience, if you have any question, you can raise hand or you can type your question in the Q&A. Okay, so I have 2 questions. Maybe I can start. So first, for the third generation of the system, it seems you already have a pretty clear spike. So can you share the status of that? And what do you think is the main technology barrier and the main solution you need to go to that much smaller volume?
Something like this?
In your comparison table.
This one. Yeah, the
third generation.
The main technology barriers are connectors and batteries. When you talk about scaling a large number of to a large number of contacts, you just cannot have these ball seal connectors. So this can put like 8 contacts. So imagine the size of this when it comes to 200 contacts or so. And this is about 22 cubic centimeter device. So you just cannot do that, although this is very reliable, very robust technology. You cannot use it for a large contact system. So I think that's the barrier number one. How can you scale contacts? Barrier number two is with miniaturization and the large number of contacts, you obviously want to have better batteries. You want to have better capacity, which I think, is a volume limiting function. And it just comes down to how often you want to recharge it. So it's kind of medium level issue. But the other thing is also, how do you want to package this? Packaging is the another big deal, right? So people have been using these titanium cans for decades. They're not necessarily very susceptible for great miniaturization when it comes to the RF coils and properties. So all those things that are, very, very, end application relevant are the things that typically people who work in the electronics completely overlook. Electronics is not the problem. Getting things miniaturized, getting things at the economy of scale that you need, that's really not a problem. It's a lot more about these mechanical issues and making sure that you pass through the full range of verification steps through biocompatibility, sterility, packaging, cytotoxicity, all of those tests.
I see. Thank you.
So there is a question from Professor Cavallaro. Is the battery pack in the controller rechargeable? There has been concern about implanting lithium-ion rechargeable, I assume induction charging like a smartphone. Yeah, so that is right. The batteries do have both primary cell and rechargeable batteries. In this particular case, we did use rechargeable battery in this one. And we have used battery that has been qualified for medical grade implants. Those batteries do have indeed quite stringent requirements. They have this 0 voltage discharge capability that you can literally drain them to 0 and do not have to deal with residual chemicals. And the company that's making these batteries for neuromodulation world, they pride themselves with their data sheets where they have shown actual 10-year testing cycles. So they basically have batteries that have started to test more than 10 years ago, and they tell people, look, we've cycled these batteries through so many cycles, recharging, we looked into all of this biocompatibility and chemical footprint, and we can say, yeah, this is really passing all of the safety regulation when it comes to medical. And that's a huge consideration. Like I like to, say my rule of thumb is for anything that FDA has not yet seen, which is, for example, new battery chemistry or something that needs to be developed like optical or something that they have not yet signed off on as the predicate device, you need to count for like 10 years, right? So anything that you need to prove, it will take you a decade to really get there and then show that it's really holding up all the requirements.
Okay, I just put a follow-up question. Your colleague Aydin Babakani has been working with some of us on pacemaker type issues, and so I'm just wondering about things like energy harvesting and is the power budget too high to do any kind of energy harvesting?
Energy harvesting to what I have looked into is not possible. I've actually done some work. You can find the publication in 2015 VLSI symposium, where we literally turned a rat into a battery. We took 3x2 thermoelectric harvesters. We put an array of these that you can put in a centimeter cubic. We put the aluminum antenna as a thermal antenna on top of it and put it all on titanium plate down below that it's biocompatible and all. And we could get like 640 microwatts per centimeter square, which would be enough to do full sensing and stimulation for something that's not like milliamp levels. But if you look into across the board, like what are the options? RF harvesting, not enough. What requires proximity for like inductively coupled, basically charging, which requires then external device. You just cannot have the holy grail is autonomous energy, right? How can you have autonomous energy? The best way to do that is photovoltaics. So if you have some kind of a photovoltaic window that you can patch into your skin and able to provide reliable supply indoor and outdoor, that would be great. But again, if you try to push that through FDA, it's going to be hard. And then piezoelectric, not enough. Ultrasound, difficult to get any meaningful levels. Muscle, very difficult. We've looked into chemical, right? If you look into the fundamental what a battery is from the chemistry, we've looked into like potential hydrogen difference between the in the body, like you have in your glands in your mouth or further down in the gut. If you can have like a relative pH difference with respect to the normal, the baseline, we thought you could somehow harvest the energy from there. Can do it. Muscle, maybe microwatt level, it's hard. So for the cardiac pacemakers, I'm also looking into some technology for these intravascular cardiac pacemakers. You gotta have something that's kind of bridge technology. You can put wireless pacemakers in the vein, but
Right.
you need to have something that's like in the pectoral area that is implanted, that serves like as an energy buffer, and then you have some external device to kind of transduce power. Harvesting alone, autonomous, it's a great question, but it's very hard. I'm just unable to get to see anything that's providing meaningful solution for that.
Okay, thank you.
Yep. So there is one more question. How promising is the Neuralink progress? DBS field recording from 100 patients, intraoperatively implant leads. 1,000 constants say promising, at the same time not believable in terms of getting good quality data. Yeah, so I would take this as a word of caution. I really like the robot and a lot of technology they've developed, but I'm just not connecting the dots on the clinical translation of this. For BCI, for animal research, it's a fantastic tool. You can do all you want to record, but like I said, and I'm sure you appreciate that from the DBS field, that there has to be ethical and clinical justification for accessing deep brain structures. And that must involve stimulation, unless you're doing epilepsy monitoring, and then you can just do localization. For the BCI, I mean, the one thing is the reliability of the insertion of these threads, which is now like 85, 90%, not the end of the world if you can't have every single one of them. But just like closing the therapeutic loop and getting anything that's deeper than the cortex would bring a lot of questions. Right now, epilepsy monitoring is the unique opportunity where we can access deep structures of the brain, but justification for inserting thousands of electrodes in human subjects, I just can't see that happening. And the other question is also, like, I think it's going to be interesting to see, and I've asked questions of Neuralink people, now that you have access to a couple thousand channels in animals, what is the learning that you can bring from those raster plots that you couldn't see from fewer channels? And what really fundamental advances that does that give in the BCI area? And then the next question is obviously therapeutic translation. How does that really go into therapy, and what indications are we able to support? And then there is obviously practical challenges of getting the robot in the operating rooms. Having seen stereotactic frames and surgeries and looking at the robot, it's hard to imagine how that can be a mainstream technology with a lot of nuances of the operations and so on. And I think NeuroPace is a great kind of scientific I think technology, but as to chronic viability, a lot of questions remain.
Okay. I think there's no other questions. Okay, so let's thank Professor Dejan Marković for the great talk.
I'm happy to engage with anyone who wants to have a follow-up discussion. I, always appreciate and like to discuss, questions and considerations with the community. So this is my email address. Please feel free to reach out. Always happy to arrange a sidebar or anything that would take this further to the benefit of society.